Good afternoon, morning or evening. My name is Piet van der Graaf and it's my pleasure to introduce you to this session on quantitative systems pharmacology in clinical development, current status and trends. Quantitative systems pharmacology, or QSP, has been defined as the combination of approaches from pharmacokinetics, pharmacodynamics, or PK/PD, and systems biology. Through PK/PD, and increasingly also physiologically based pharmacokinetics, or PBPK, we aim to obtain confidence in the compounds, building the three pillar foundations of target exposure, target engagement, and target modulation. The focus of systems biology, on the other hand, is to build confidence in the biological targets and mechanism through quantitative models of pathways, pathophysiology, and disease. QSP has gained significant traction over recent years, not just in industry, but also at regulatory agencies, as illustrated by this analysis from FDA, which shows the rising number of submissions supported by QSP over the last decades. Interestingly, in this analysis, FDA showed that the majority of these submissions, almost 75%, were focused on clinical development in phase 2 and 3. The objective of this webinar is to share with you some case studies to illustrate what such applications of QSP in clinical development looks like, what its impact is, and also where the field is going in this area. One example where QSP has become an integral part of drug development and regulatory submissions are bispecific monoclonal antibodies, specifically in immuno-oncology. At Certara, we have supported dozens of programs with QSP modeling, and it has almost become an expectation for the justification of human dose selection. In a recent example, we supported an IND submission for a bispecific, and our client feedback was that they had submitted another IND for a very similar compound 6 months earlier, and that application of QSP had saved them 2 dose levels in the first human study. We have developed very similar approaches for other novel modalities such as PROTACs and gene editing ligands. This slide shows some examples of the various modality, disease, and pathway QSP platforms we have developed over the years at Certara. As you will see later, we now routinely and seamlessly integrate these platforms with PBPK models of both drug and biomarkers to guide clinical trial design. The 2 case studies we will present shortly are examples of applications of our platforms for inflammatory bowel disease, IBD, and Alzheimer's disease, AD, highlighted in red here. We selected these case studies because they illustrate 2 different but complementary approaches to link QSP models to clinical endpoints. What all QSP models have in common is that they predict modulation of biomarkers in a detailed mechanistic manner, where the definition of biomarkers is of course very broad, and in principle includes all biological species in the model, whether they are clinically measurable or not. For certain diseases, like Alzheimer's, we have sufficient understanding how to link the biomarkers generated by a QSP model to certain clinical endpoints in a mechanistic manner. My colleague, Hugo Gertz, who heads our Neuro-QSP group, will illustrate this approach with a case study where we supported the clinical development of a recently approved amyloid antibody for Alzheimer's disease. In other areas, like for example inflammatory bowel disease, this fully mechanistic approach is not possible, often because the clinical endpoints can be somewhat arbitrary qualitative scales, such as the Mayo score. For these situations, we have developed an approach where we can link the outputs from a QSP model to clinical endpoints using AI and machine learning. At this point, it's my pleasure to introduce my colleague, Doug Chung, Senior Director QSP at Certara. who will present the first case study illustrating this novel concept.
Hello, everyone. I'm excited to present to you a very promising recent approach that we have developed. The title of my talk is Predicting Clinical Endpoints Using QSP and Machine Learning, a case study in inflammatory bowel disease. It starts with one of the most comprehensive QSP models of IBD. This captures the key inflammatory dynamics in the gut mucosa, including the effects of the cytokines, such as TNF-alpha, which is responsible for activating macrophages, dendritic cells, and neutrophils. The model also contains more recent findings for the effects of IL-23 on T-cell differentiation and activity. The model is based on a mechanistic, multi-state mathematical model that was recently published in CTS. The graphic here shows the full list of key biological compartments, cell types, and cytokines in the blood, gut, and lumen. We have since expanded the model to include damage-associated molecular patterns that are the result of damaged epithelial barrier function and the resulting necroptosis and inflammation. The model can estimate clinically measurable biomarkers, including fecal calprotectin and serum C-reactive protein. The IBD QSP model simulates both Crohn's disease and ulcerative colitis using the same underlying biological mechanisms. The goal of this model was to answer three important questions. First, what is the right target? In our example, we investigated the effects of inhibition of the TNF receptor and the IL-23 receptor. Second, what is the right dose? We validated the treatment response and explored dosing regimens for an anti-TNF-alpha and an anti-IL-23 biologic for both induction and maintenance dosing. And third, what is the right action? We explored differences in treatment response for virtual patients of varying disease severity and varying CD4 T helper cells type 1 and type 17 activity. But to have real clinical impact, we developed a machine learning approach to predict the clinical scores using simulated gut markers from the QSP model. As you may know, the clinical scores for IBD are composed of subjective questionnaires, and there are no known mechanistic links between the gut biology and these scores. Now, in the next few slides, I'll share our promising approach in predicting these clinical scores. Now, before we could connect the gut inflammatory markers to the clinical scores, we needed to better understand the possible relationships between them. So we conducted an extensive literature review to find known correlations between the gut cytokines and immune cell activity and the clinical scores. We found that there are strong correlations between the cytokine levels secreted by CD4 T cells, antigen-presenting cells, macrophages, neutrophils, and the clinical scores, such as the total Mayo score and the CDAI score. Unsurprisingly, T helper type 1, macrophages, and neutrophil activities are positively correlated with higher clinical scores and disease severity. Interestingly, T regulatory cell activity has mixed correlations with the clinical scores, which highlights the need to understand possible differences between locally proliferating and recruited T regulatory cells. Using these correlations, we then search for mechanistic evidence linking these gut inflammatory markers with the clinical biomarkers and the scores. For example, there is strong evidence for the role of interferon gamma in vascularization and the role of neutrophils and subsequent release of calprotectin. We selected a focused list of key cytokines and immune cells to serve as predictors for the clinical scores, which serves as a basis for our predictor. So we wanted to be able to predict the change in clinical scores in response to anti-TNF-alpha and anti-IL-23 treatment to better understand the mechanisms that affect rates of response and remission. So we built a multinomial logistic regression model mapping the simulated gut inflammatory markers to the clinical biomarkers, in this case, fecal calprotectin and serum CRP, and then to the clinical scores, including the Mayo endoscopic score, the total Mayo score, and the CDAI score. Using this focused list of predictors, we trained our model using the validated virtual population from the model publication and the literature data establishing the relationship between the clinical biomarkers and the clinical scores. So that means that for individual virtual patients, we can input the simulated gut cytokine and immune cell levels into the MLR predictor to compute the probabilities for each subscore. For example, we simulated a severe colitis patient under 4 different treatment scenarios: untreated, treated with adalimumab, treated with mirikizumab, and combination therapy. We predicted a drop in the Mayo endoscopic score from severe or 3 to a 50-50 chance of a score of 0 or remission, or a score of 1 or clinical response following the combination therapy. We are able to compare treatment response between different monotherapies and combination therapies for the same individual patient. Predicting the treatment response for individuals is good, but we can do better by applying the same approach to an entire virtual population to guide clinical trial design. We asked the question, what if the same group of trial participants were given an increased dose or a combination therapy? We conducted a virtual retrospective study using published clinical trial data by first replicating each trial by building separate virtual populations that match the baseline distributions that are distinct to each trial. We then simulated an alternate dosing regimen. For example, in the recently completed VEGA trial investigating a combination of an anti-TNF-alpha and anti-IL-23 therapy, we predicted that if we increase the maintenance dose amount, the percentage of patients classified as clinical responders would increase 14%. We evaluated several colitis trials and predicted that the percentage of clinical responders could have been increased significantly. To summarize, we built a comprehensive IBD QSP model capable of exploring biological mechanisms and treatment targets. We connected gut inflammatory markers with the clinical scores based on extensive literature review. We built a machine learning approach to predict the treatment response for an individual patient. We applied the clinical score predictor to actual trial data to be able to guide clinical trial design. We feel this approach could be applied to other disease areas with the right combination of QSP modeling and robust clinical biomarker and endpoint data. Thanks for your attention. I would like to introduce my colleague Hugo Geertz, Head of NeuroQSP at Certara, who will present the second case study.
Thanks, Doug. Next up is predictive QSP model for a case study in Alzheimer's disease. Alzheimer's pathology is characterized by 3 different pathological processes: amyloid formation in amyloid plaques, neurofibrillary tangles around the protein tau, and neuroinflammation. In this case study, we will actually combine those 3 pathologies and combine it with computational neuroscience that allows us to predict the kind of the functional outcome in those patient populations. First up is the amyloid aggregation pathology. Here we actually show a model that has been published a couple of months ago together with our clients at Eisai, in which we combine seamlessly a PBPK model for lecanemab or any other biological antibody with a mechanistic QSP model for amyloid beta aggregation and synthesis aggregation. As you can see, we have different forms of A beta species in which we have a biophysically and informed mechanistic model that drives the aggregation between the different species. Along the way, we are able to actually predict the count of the CSF plasma biomarkers and the imaging PET amyloid biomarkers as well, and also the ARIA-E side effect, which I will come back to later. Because the neurotoxicity is driven by some of the intermediate species which are not accessible to measurements, this model allows to actually connect the biomarker dynamics to a kind of functional outcome, as I'll show later on. This model can actually be used for a number of applications: therapeutic index, maintenance doses, and patient selection. So the insights that the model generated actually and predicted was twofold. On one hand, we were able to actually show that lecanemab was superior in driving the decrease in protofibrils, as you can see after 1.5 years of treatment, compared to the other antibodies, aducanumab and gantenerumab. And it turns out that this proportional decrease in protofibrils was associated with a proportional improvement or a greater reduction of cognitive worsening in the clinical outcome. We were also able to actually then use this model to predict the outcome of the more recently antibody donanemab from Lilly. The other major insight that the model generated was the kind of the ARIA-E side effect. ARIA stands for amyloid-related imaging abnormalities, which actually corresponds to a kind of microbleeds in the cerebral vasculature, and is really hampering the kind of the rollout of that therapeutic modality. So we were able to show in this model that aducanumab had a much higher reliability than lecanemab, and that donanemab was kind of intermediate in between the two drugs there. On the other hand, we were also able to develop a time-to-event modeling approach that allows us to optimize the titration schedule for novel therapeutics to mitigate the risk in those patients. This slide shows the tau QSP model that is again a combination of PBPK model for tau antibodies and the interaction of the antibodies with the tau that's being secreted from one neuron into the other neuron, which is a kind of the current hypothesis of tau propagation. We used the kind of the appropriate compartment volumes because this synaptic volume is very, very small. And therefore, we used the kind of a tortuosity of the antibodies to show the following results. We were first able to show the kind of the phase 1 prediction for Biogen's gosceneuramab that showed the complete target engagement of the free monomeric CSF tau. But on the other hand, the uptake of seed-competent tau in the afferent neuron was not reduced by gosceneuramab, which could explain the phase 2 failure in PSP and Alzheimer's disease. If you would actually use a hypothetical gosceneuramab type antibody that is more selective for seed-competent tau, we will be able to see a kind of an effect there. So this approach would actually be helped to optimize the pharmacology of novel anti-tau antibodies. Of course, the big payoff comes when we can combine those biomarkers with functional clinical outcomes. Now, human behavior is driven by activation of certain neuronal circuits in the human brain. And therefore, we used computational neuroscience, which is basically the simulation of action potential dynamics in complex neuronal networks. As you can see on the right side, this is a model for memory trace stability that we used here. And beta amyloid affects ligand-gated ion channels like glutamate and nicotinic receptors, whereas tau affects voltage-gated ion channels like sodium and potassium channels. So this is a way in which we can bridge the molecular biomarker changes in amyloid and tau to a more functional clinical readout that have been calibrated using the ADAS-Cog or the CDR sum of boxes. This gives us the kind of the possibility to look at virtual twins, so in which we would develop an in silico model of an identical patient based on the baseline variables, co-medications, genotypes, amyloid load, and tau load, and then simulate the trajectory of the placebo form of those patients over time. At the end, then we can compare on a per patient basis the effect of the treatment between the actual outcome in the trial and the in silico trajectory of the placebo patients. This is very helpful for trials with invasive gene therapy in which we cannot really use placebo patients, or to revisit small phase 2 proof of concept trials that had inconclusive results because of the small number of patients. To wrap it all up, this is our kind of the integrated Alzheimer QSP platform, in which we now are combining the different boxes and modules. So again, all the way from a PBPK model in box 1 to the amyloid aggregation model in box 2. It is of interest to note that we have also developed a neuroinflammation model, more specifically a microglia model that focuses on the TREM2 pathway, the RIPK1 pathway, and the inflammasome pathway, and is driven by the amyloid aggregation platform as a pathological event. The A-beta monomer dynamics are then fed into the neuronal network firing that drives the tau release and the tau spreading model. And ultimately, both of the amyloid beta and the tau will actually then converge into the kind of the computational neuroscience network to generate a kind of a prediction of the functional cognitive readout. As you can see, we have a different biomarkers in red: amyloid PET, amyloid biomarkers, tau biomarkers, and tau PET imaging biomarkers. And the ultimate combination will result in a functional cognitive readout. This allows for optimizing trial design for combination therapy and explore the personalized therapies on functional outcomes as well. That's all. Thanks a lot for the attention.
Thanks, Hugo and Doug, for sharing this great work from you and your teams. And of course, we would also like to acknowledge our pharma partners on these case studies, Eisai and Sanofi. Well, I hope these examples have given you a good idea how QSP modeling can be linked to clinical endpoints and therefore can be used to inform decision-making in clinical developments. The examples were chosen because they represent two different approaches. And in the next few slides, I will show you how we are now integrating these to develop the next generation of QSP platforms. Although today's focus was on application of QSP in clinical development, that does not mean, of course, that QSP cannot also impact earlier stages of drug discovery and development. In fact, we believe that investments in QSP cannot start early enough, and that a QSP platform is a knowledge and data repository that develops alongside a drug development program to address different questions at different stages. For example, our IO simulator platform, which we did not cover today, is now being modularized into submodels that can either be used in a standalone manner, for example, to guide target selection in early discovery when there may be very little data available, or to guide biomarker research. But at the same time, these modules can be integrated when more data becomes available and the questions shift towards clinical application. We believe that such a platform approach to QSP that is fully integrated into a drug discovery and development program requires a bespoke in silico environment that brings together modelers and non-modelers, and drug developers and regulators. Until very recently, such a tool was not available, but I'm very pleased to announce that with the publication of this paper in ASCPT's PSP journal, QSP Designer has arrived to fill this gap after 7 years of development. If you're interested to learn more, the tutorial, which includes a video demo, is available to download for free from the PSP website. There will be a webinar in a few weeks' time, which is now open for registration. A full workshop will be given at Page next month. And you can request a free trial license from simsip.support@certara.com. In my last two slides, I would like to give you a view of how we are now integrating the QSP Designer platform with other in silico technologies, which we believe is defining how QSP will be used in clinical development in the near future. First, based on the very successful proof of principle Doug showed for IBD, we are now linking many more of our QSP models to our Codex platform, which contains very rich clinical data sets for some 60 diseases. In parallel, we are now deploying state-of-the-art AI and machine learning technology and platforms to make the link between the QSP models and clinical endpoints even more robust and predictive. So, this is what the future of QSP in clinical development will look like. But I should emphasize that the future is already here, and this workflow is already being implemented in actual projects. We would, of course, be more than happy to discuss this in more detail with you. Thank you very much for your attention, and we look forward to your questions and comments for the panel discussion that will follow now. Hello, my name is Piet van der Graaf. I'm a Senior Vice President QSP at Certara, and it's my pleasure to be the moderator of this panel discussion on application of QSP in clinical development. And it's my real pleasure to introduce to you our two panelists, Hugo Geerts, who heads up the Neuro QSP group at Certara, and also Doug Chung, Senior Director, QSP Consultancy at Certara. So we've already got quite a few questions, but please keep posting your questions for our panelists. And let me start, Doug, with the first clarifying question from someone who asked that you mentioned the term MES in your talk as one of the clinical scores in IBD. Could you please clarify what that stands for?
Yeah, so MES stands for Mayo Endoscopic Score. We started with this particular score because we felt it was the most mechanistic and at least subjective out of the clinical scores for colitis and Crohn's disease.
Okay. Can you maybe explain a little bit more what the Mayo score actually entails? Because I think that really helps to understand why we had to use a AI machine learning approach rather than a mechanistic QSP model to kind of link the biomarkers to those scores.
Sure. Yeah. So the first point I'll say is that the Mayo score is generally used as the gold standard in terms of clinical endpoints for IBD trials still at the moment. And it's composed of a series of questionnaires, both for the physician and for the patient, as well as maybe some less subjective kind of blood-based measurements. So we felt that the AI ML approach was appropriate to try and connect the gut level inflammation to these subjective scores.
Okay, excellent, thanks. And then maybe, Doug, can you explain a little further how the predictions particularly of the simulations you showed at the end, where you kind of showed virtual trials of ideas and combinations and dosing regimens that hadn't actually been tested yet. So how were those predictions verified or, well, even validated?
Sure. I'd say the first step of verification we did was with the publicly available data. So for trials that have been completed and published, we're able to match the baseline distributions for current biomarkers such as fecal calprotectin and serum CRP, as well as other kind of baseline measurements for the study. What we cannot currently verify is the unseen trials that we simulated with. So the alternate dosing regimens, of course, it's if we could, we would gather the same group of trial participants from that published trial and administer a different arm, a different dosing. But since that's not possible, these are more prospective or predictions that the model offers. Thanks.
Well, I'm sure we'll come back to the discussion around QSP model verification and validation, and I'm sure there's more questions on that in the audience too. So please keep posting your questions in the chat box. But we have several other questions for now. So Hugo, shifting towards the Alzheimer work that you showed. Quite impressively, I think you showed that the QSP model actually predicted the clinical success of lecanemab more than 6 months before the trial data were actually there. And so based on that success, how is the model now being used in other amyloid antibody programs and also perhaps novel mechanisms such as tau and neuroinflammation?
Yeah, I think that was one of the insights that the model generated in terms of differentiation between lecanemab and the other antibodies that have kind of failed or semi-failed in clinical trials. So based on the hypothesis that these protofibrils, the soluble protofibrils, were the kind of neurotoxic agents, we showed that the pharmacology of lecanemab was optimized to actually reduce that amount of toxic protofibrils to a much greater extent than the other antibodies. So I think that's one issue that we could actually predict with the model. The other important insight was the kind of the side effect liability, the area E, which is at this moment the kind of the major hurdle for uptake in the market, major safety hurdle. Again, here, based on the pharmacology of lecanemab, we were able to show that the liability, the frequency there was much lower. So Going forward, I think that's it shows that we can, let's say, predict the kind of both the efficacy in terms of reduction of worsening of the cognitive symptoms, as well as the ARIA-E side effects for any novel medication that affects the amyloid aggregation pathway. Not only antibodies, but also small molecules that act on, let's say, the synthesis of A-beta from the amyloid precursor protein itself, like gamma secretase modulators, which are now kind of the new idea there, and give them a kind of an idea in terms of both the efficacy and the side effect. For amyloid antibodies, the model allows you to optimize the titration schedule to actually maximize the efficacy over the side effect aspects. And even going forward in terms of what happens in the market when a number of those antibodies become available is the kind of treatment guidelines. So one of the ideas that we currently will present at the upcoming Alzheimer meeting in Amsterdam is a simulation of different patient profiles and their optimal titration schedule, the time at which the amyloid load will actually reach negativity, the maintenance schedules, and so on. So I think this is another application of this very important platform.
No, I certainly think that the whole question about differentiation, particularly for amyloid antibodies, will all be around the balance between efficacy and side effects, and particularly ARIA-E. And I think that's a great example of application where you can really now use the model to optimize the dosing schedules. So maybe back to the previous question that someone in the audience asked to Doug about how do you validate these QSP model predictions of trials that you haven't actually done yet. Now you were actually applying this in a real program, right? So you were actually working with a clinical team that was already in late-stage clinical development, and so how did you persuade the clinical team that the model predictions that we retrospectively now knew were actually giving a pretty accurate prediction of a trial 6 months before the results? How did you work with the team to build that trust and confidence?
Sure.
Yeah,
Yeah.
I think it's, go ahead. Yeah.
So should I start? Yeah. I think it was very important just to involve the team from the very beginning, right? So they're involved in the process of building the biological map and seeing the predictions to the established clinical trial data that's already out there. We are waiting on some feedback. So for certain clinical trials to complete in order to get a better picture of the extent to which we can verify these models. But I think as time goes on, these trials and other related trials will start to complete and publish their data. And at that point, we can start to verify to a further extent. But definitely involving the client from the beginning and seeing all those results. And something I couldn't share today was, of course, some of the additional programs the client was running that we're able to verify as well. So,
right. Okay. Thanks. Hugo, any further comments on your experience on building the trust in a clinical team?
Yeah, I think in our case with the amyloid aggregation project that we had, I think the trust was based on the fact that we had a very detailed biophysical mechanism that we built into the model rather than a kind of a more phenological state transition scheme like many other models had done in the past. And so we were able to actually show, for instance, the natural history of amyloid biomarkers, which actually reflected quite well the kind of the observed longitudinal studies in the field. So, and also we worked quite extensively with the whole team at ESA, including also the biomarker team. That was very helpful in actually help us to set the parameters and the concepts of the model.
Okay, well, we're getting a lot of questions, so I'll keep going through the list, and we'll try to cover all of them. If we don't manage to go through all the questions in this live session, we're very happy to follow up with you and provide you answers afterwards. So next question is maybe Doug, I'll start with you. So how was QSP linked to AI, and then building all that, is QSP expected to be transitions to become part of AI? Or maybe I'll add the follow-on question, or maybe the reverse. What do you think?
Yeah, maybe I could just speak to the case study that I showed here today. So we felt that when there is a lack of understanding, a mechanistic understanding between the connection of the local gut biology to the actual clinical endpoint, we think AI or ML could be a nice tool to try and bridge the gap between the two. So there's extensive data on the clinical biomarkers that are measured in the clinic, but there is no connection between the two. So we had to try and find an approach here. So in general, what happens is the QSP model provides simulated biomarker data, and that feeds into an input which serves as a predictors in a AI or ML approach. And then in an iterative fashion, we need to verify that the predictions of the AI model are aligned with the predictions of the QSP model and ultimately to the clinical trials themselves.
Yeah. So as we try to explain, we very much see the partnership between QSP and AI as the key to success. And we think it's really the synergy between the two that will kind of define the next version and generation of QSP models, and particularly because we think QSP models are incredibly strong at building mechanistic models of biomarkers. Linking those biomarkers to clinical endpoint is often challenging, mainly because of the nature of the clinical endpoint, as Doug illustrated in the IBD case study. But Hugo, you showed the alternative approach, where actually we can link the biomarkers to clinical endpoints in a fully mechanistic manner, as you showed in the Alzheimer case study. Now, can you see applications of AI and combinations of AI and machine learning with QSP in neuroscience?
Yeah, I think certainly. I think even if the current approach that we have done so far is actually based on the observation that human behavior, which ultimately drives the clinical skills in cognition and behavioral disinhibition and so on, is driven by activation of neuronal circuits, kind of action potential dynamics and so on. So I think one of the applications of AI and machine learning is to really figure out how the signature of action potential properties, either in terms of oscillations or network dynamics, might actually be related to the clinical symptoms. And we have a lot of clinical data in which subjects are being studied with EEG or magnetoencephalography, or even Bolt fMRI. So a lot of biomarkers that report on the activation of neuronal circuits and correspond to a certain clinical outcome. So I think that's the way how we see AI actually coming into getting a better relationship between the simulated dynamics of action potential properties and the corresponding clinical functional outcomes.
Yeah. Okay, so next question, and maybe I can try and answer that, is that the value of QSP modeling is growing in both development and discovery. Now, the cost of this can be easily absorbed by larger pharma, but are there ways that smaller companies, maybe even startup companies with limited funding, can utilize QSP in discovery and development? So I think the answer to that is yes. And actually, we partner with many smaller companies, biotechs, even startups. Now, the reason we can do this is twofold. One is that we, as we have shown, have developed platform models, because I think the question is not just about the cost, but also the time it takes to really develop such platform models. Now, because we have been investing in building these platform models over many years, as Hugo, for example, showed in Alzheimer, we can now actually use them and generate answers. In the case of, for example, Alzheimer amyloid antibodies, as a matter of weeks sometimes. So we can really kind of utilize the prior investments in development of these platforms to move real fast. The other thing is, of course, that QSP modeling comes in many flavors. The example I showed you of the area where we apply mechanistic QSP modeling in the field of bispecifics is an example where the models are actually relatively small compared to the big platform models that we showed in the case studies. So here, actually making such models bespoke for a particular asset, again, can be done in a fairly short period of time. So next question is around how much can QSP modeling be used in the field of rare diseases, particularly where initial trial subject numbers are much lower to validate the prediction. So I think, Hugo, I'll start with you and then move to Doug, because I think you've both worked in rare diseases and applied QSP models. Hugo?
Yeah, I think that's a very important question. I think in the sense that in many of those rare diseases, we don't have much data to rely upon. So I think that's where the concept of the virtual twin, which is identical to the kind of the patient in the field with all the baseline characteristics, is an important starting point to really develop a model for the longitudinal trajectory based on the insights of the pathology. And then, so one of the ideas that we're currently developing is to have this virtual placebo twin model in which we would actually simulate the trajectory over time of the patient with identical characteristics as the actual patient in treatment, and then do a kind of per patient comparison there. So I think that that's the way in which QSP can actually be quite helpful in supporting clinical trials for rare diseases.
Yeah, I think the use of virtual twins as synthetic external controls is a really exciting area where QSP can have a big impact, and was actually a topic of discussion in last week's workshop from University of Maryland and FDA, which was specifically focused on application of QSP modeling in rare diseases. Doug, any further thoughts on this topic?
Sure, yeah, maybe just thinking further ahead into the future. I just attended the past conference at ASGCT, and there's a lot of conversation around how modeling could possibly help with the current backlog of applications at the FDA. I think the real challenge is connecting protein level biomarkers to actual clinical endpoints such as motor function and so forth. In the case where, maybe we can't build that bridge yet at the same time, there's not enough data from limited trial participants to build a full AI ML approach. But one possibility is if related diseases can combine their data in a pre-competitive environment, there may be an opening to start applying some AI ML approaches if the diseases are related enough and there's enough mechanistic validation to go with this approach. So maybe a bit far off in the future, but could be promising.
Okay, thanks. So another question for both of you, maybe Doug, you can start. Are the models that you presented based on individual data or on mean data?
So it's a mixture of individual and mean or summary statistics type of data. The majority of the published clinical trials we show you here today are all based on summary data, but there was some individual data we had access to, both in publishing and within the client data that we are working with.
Right. And Hugo, is that the same in the applications in neuro?
Yeah, so most of the applications in neuro and the first validation and calibration have been done using summary statistics of published clinical trials in Alzheimer's disease in the past. But we have done some examples in which we actually looked at individual patient trajectories of a placebo arm in an Alzheimer trial, in which we had individual characteristics of patients in terms of their co-medication, smoking status, and genotypes, and then simulated the kind of the longitudinal trajectory over 24 months in a kind of placebo situation. So yeah, it's a mixture of both of the summary statistics and individual data that we had.
And obviously the concept of virtual twins, particularly when it is applied in rare diseases, becomes almost the ultimate use of individual data, I suppose.
Indeed, yeah.
So I think we may have time for maybe one or two more questions. Probably one. So maybe Hugo, briefly, you mentioned that PBPK is now also used to model not just pharmacokinetics of drugs, but also the reverse of distribution of biomarkers. Can you, in like 30 seconds, maybe briefly explain how that works?
Yeah, it's a kind of modeling of the same concept, but done in the reverse direction. So most of the, let's say, CNS biomarkers or proteins or peptides generate inside the brain and show up in the CSF and the plasma. So we optimize the PBPK model to actually correspond to the kind of clinical data that have been observed in longitudinal studies. And that actually was the basis of some of the biomarkers dynamics after therapeutic interventions.
Thank you. Okay, well, I think that's all we have had time for. As I said, we're happy to follow up with anyone if there's more questions. Thank you, Doug. Thank you, Hugo.